Hello, I'm Kenneth Eck with the Du Bois County Extension Service, and today we'll be talking about nitrate testing in standing corn. In this year of 2012, we've had severe drought conditions in most of the Midwest, and with that, concerns for nitrate accumulation in the stalks of corn and other related species. That nitrate, if left in the corn and not identified beforehand, can cause problems in livestock that may eat the corn, shucks, stalks, leaves, and other parts of the crop. Today we're going to talk about some basics of nitrates, some ways we can test for it, and perhaps things we can do to kind of correct it before it gets to our livestock in the local feeding system. One of the things we'll be talking from today is the publication Utilization of Drought-Stressed Corn, which is available through the Du Bois County Extension Service here in Jasper, Indiana. To start with, we're going to talk about nitrates in general. The reason nitrates are toxic comes from the fact that the nitrates, once they get into an animal system, can change hemoglobin, which transports oxygen throughout the animal system, into methaglobin, which does not transport oxygen nearly as efficiently. The result can be problems with the livestock's health, including tremors, heart rate and respiratory increases, changes in color of the animals to a bluish or a chocolatey brownish type look, staggers, and eventually suffocation as oxygen becomes less and less prevalent in the animal's blood system. To do the testing in a field, there's two different tests we can utilize to identify nitrates in standing corn. The first is called a quick test, which I'll be demonstrating today, in which sulfuric acid is used to get a very quick qualitative analysis of what's in the cornfield. By qualitative, we mean we simply identify whether or not nitrates are in the corn and at a level that may dictate further testing being needed. The second test is laboratory analysis, or quantitative, in which a sample of corn, usually chopped corn of about 1 pound, is put in a brown paper bag and submitted to a local laboratory through a feed mill, through the Purdue ADDL, or Animal Disease Diagnostic Laboratory in southwest Indiana or on the main campus, or some other test lab capable of doing a quantitative test to tell exactly what the amount is in that corn crop. For today, what we've done is basically already selected some corn from the field. To do that, we're going through several places in the field where we might think there are high places and low places that may be of concern. Again, the concerns we have out there is that this corn plant, although it does have ears on it, was not able to utilize the nitrate taken up from the soil. Again, that nitrate may be from over-application or very well-fertilized fields with nitrogen, or maybe also from having a lot of manure applied, which is common in this county. During drought conditions, the leaves are not able to process the nitrogen or nitrate as easily, and because of that, it starts to accumulate in the stems and stalks. Once the plant becomes back to normal again because of lessening of the drought conditions, the temperatures lowering, or rainfall picking up again to where the plant can go back again and use the nitrogen in a way it should to convert it to needed parts of the plant, the nitrate problem may be reduced or actually go away. But until then, we have to test for it to make sure there's not a problem with feeding these kind of stalks to livestock. What we're going to start off doing is, first of all, go through and collect samples again from areas of concern and more normal areas in the field. We'll come back and strip off all the leaves from the crop, so all we have left is the stalk itself. Once we're done doing that, we're going to take a knife and strip the stalk so we have only the pith showing in one half of the stalk itself. At that point, we're going to start using sulfuric acid, which is a major concern as far as getting it on your skin, clothing, or other parts of this experiment. So we're going to be using goggles and gloves to make sure nothing happens to me while I do this. Again, just a reminder, this test is not a quantitative test. What we're going to do is we're going to locate different color changes within the stalk, if there are any, which indicate nitrogen being present in the stalk. We're not usually concerned as much about it being in the leaves, where it'll be a lot lower than the stalk itself, or especially in the grain or the tassel, where it's very rarely found. Again, it's going to be accumulating, if anywhere, in the stalk itself. So that's what we're looking at mainly for this test. For this test, we've split two different stalks from two different fields. We're going to apply sulfuric acid again to the stalk in between each of the nodes in the pith area and let it sit for roughly about a minute. After that minute, we'll be able to see what changes have taken place to that corn stalk as far as color goes, and then compare it against a chart we have developed by Purdue University and other universities to tell us where we may have concerns in these corn crops. Bluish black look to our corn stalks when we're done, indicate nitrogen. In most cases, we're going to find it probably in the lower parts. That's where it's going to be the highest concentration at the beginning, where it's being taken up by the plant. And as the nitrate problems become less of a concern, we'll see a lightening in color to almost a yellowish or almost a clear color as we go farther up the stalk, if there is not a problem with nitrate poisoning all the way to the top. Crop we have right here, we can see there's very dark areas indicating there is nitrate in this crop behind me, and it's going up as far as I've tested it. In a normal circumstance, I'd go ahead and put crop all the way up to the top just to find out exactly where we might have concerns at, how much this crop might be usable. The same also occurs in this second stalk, also taken from this field. Third stalk that comes from a field nearby here is showing that nitrate is not a concern. Basically, we do have a little bit of color change here, but it's mainly browns or light yellows or blues that are kind of mixed in with greens and yellows. So basically, this stalk I would not have much concern with in making a recommendation to a producer ready to feed this to beef cattle of having a problem with chopping that. For this one, we'd probably want to raise the bar as much as possible on the chopper to try to get as high up above this area as we can. In some cases, that's not always possible. We might have it farther up, so we'd be more concerned about maybe going out, taking samples and sending them to a certified lab to see exactly what the amounts are. And then once we come back with those certified amounts, we go through and check them against charts we have that would tell them what amount of feed they would need to mix with that. Basically, we're going to try to dilute the feed with other feed sources that will not have nitrate problems. It might be a hay, it might be glutens or other things available from local mills. The other concern we would have is that when we come back, if we make silage from this, the fermentation process itself will actually reduce the amount of nitrate found in this corn from 40 to 60%. Usually a rule of thumb is 50% or half of what the rate is when it comes back from a certified laboratory with a laboratory quantitative amount. So again, there are some options that we can hopefully have in the future. Reduce it by the fermentation process and then dilute it further when it comes back from being ensilaged after 3 minimum weeks, and then use that then to mix with feedstocks. They'll be, again, reducing it as much as possible. 3 other options that farmers also use with corn like this is also doing green chop, which does not reduce the nitrate amounts as much as the silage process does. Matter of fact, it doesn't reduce it at all. Also, the concern that the nitrates can turn into nitrites, 10 times more deadly if it sits on a wagon overnight. So be very careful with that concern. A third option is utilizing corn hay, going through and chopping this into large round bales. But again, the problem there is without being ensilaged, we're not reducing the nitrate amounts in the corn stalks themselves. So for this crop, whatever we come back from laboratory analysis with is what we'll be stuck with when we feed the cattle for the most part. The fourth thing is, a lot of farmers have been asking about what can we do with the stalks after we've harvested the grain. We have grain in this cornfield right now, but we also have nitrate problems in the stalks. If this is grazed afterwards, a lot of times corn will be selectively foraged by the cattle. They'll only eat the leaves, the tassels, the cobs, things like that. And most of the nitrates, again, will be in the corn stalk itself. So if cattle are not forced to eat the corn stalk and allowed to pick what they might want, the concern there is very much reduced. Same with feeding hay. If we can feed the hay bale, but do it quite liberally, Do it in a way that the cattle will be able to pick through. They'll usually leave the corn stalks behind, again where the nitrate concern is highest, and they'll mainly eat the stalks, corn kernels, things like that are going to be less likely to have nitrate concerns. So with that, again, I'd recommend you to consider looking through the utilization of drought-stressed corn available at the Du Bois County Purdue Extension website, and get more information if that's of concern with your crop here in Du Bois County or in other parts of the state. Thank you.